Hello, everyone, and welcome to this new webinar being brought to you from the CRISPR Journal and Gen, entitled Therapeutic Advances Using In Vivo CRISPR Genome Editing. This webinar was made possible through sponsorship from TriLink Biotechnologies. TriLink Biotechnologies, part of Maravai Life Sciences, is a CDMO helping life science leaders and innovators overcome challenges in the synthesis and scale-up of nucleic acids, NTPs, and mRNA capping analogs with scale-up expertise and unique mRNA production capabilities, including its proprietary CleanCap mRNA capping technology. TriLink continues to expand its cGMP and general mRNA, oligonucleotide, and plasmid manufacturing capacity at its new global headquarters in San Diego, California, to support therapeutic, vaccine, and diagnostic customers. I'll be your host for today's event. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which is celebrating its 40th year, bringing you the latest information on the most cutting-edge tools and technology that life scientists need to keep their research moving ahead. So as scientists, it's really in our nature to be skeptical, especially about things that are touted as, quote, game-changing or, quote, revolutionary. We often hear these terms thrown around only to have things fall flat when they're put under high scrutiny. So when CRISPR burst onto the scientific stage about a decade ago and was being hailed as having a potential to cure a vast array of diseases, many investigators gave a collective eye roll. However, much of the skepticism quickly subsided as the experimental data kept flooding in, causing investigators to switch from uncertainty to optimism, and wondering how quickly we could see this technology get into patients. Well, this past June, we finally had the answer to that question, as Intellia Therapeutics, along with its partner Regeneron, announced positive interim clinical data from a phase 1 trial evaluating its CRISPR therapy NTLA-2001 in patients with a rare liver disorder called hereditary transthyretin amyloidosis, or ATTR, with polyneuropathy. This major achievement supports not only the safety and efficacy of in vivo CRISPR genome editing in humans, but also shows its meaningful therapeutic benefits, something we're going to hear a whole lot more about from our presenter in just a few moments. But first, let's meet and learn a little bit more about our distinguished guest for today, who is here to tell us about how these CRISPR therapeutics work and what some of the success means for the future. Dr. Laura Sepp-Lorenzino is the Executive Vice President and Chief Scientific Officer at Intellia Therapeutics, where she oversees research and preclinical development. Dr. Sepp-Lorenzino has expertise in oncology drug discovery and development and has had an extensive career in the biopharmaceutical industry, taking up leadership roles in some of the biggest companies with some of the most active projects. Laura's vast knowledge and expertise is highly sought after, as she resides on numerous scientific advisory boards and boards of directors. Today, Laura is going to tell us more about Intellia's progress with both in vivo and ex vivo genome editing approaches, as well as discuss the recent advances with NTLA-2001. Immediately following Laura's presentation, we'll have our Q&A session. And I want to remind the audience to submit questions so that we can have a very lively discussion. To do that, it's really simple. All you need to do is type your question into the Ask a Question box and hit submit. Now, I hope everyone takes a minute to send a question for the Q&A, but let's get to why you're all really here. Laura, the floor is yours.
Well, thank you so much for hosting this webinar and for the opportunity to share with all of you what we're doing at Intellia. These are really exciting times for us, for the field, for patients. The promise of genomic therapies, and particularly of CRISPR, is now. So exciting days. Please make note of our forward-looking statement. At Intellia, we're pursuing a full-spectrum strategy by harnessing the power of CRISPR-Cas9 to develop potentially curative therapies for severe diseases. For this purpose, we've set out to build a deep and broad toolbox of both gene editing as well as delivery solutions that will allow us to choose and deploy the right combination to make a drug product that's going to be safe and efficacious and will have all the drug-like attributes that's required for a medicine. We apply our differentiated modular solutions across both in vivo and ex vivo therapeutic applications. So for genetic diseases, we deliver CRISPR-Cas9 in vivo by a systemic IV administration of the drug, the CRISPR machinery. For immuno-oncology and autoimmune diseases, here we use CRISPR as a tool that allows us to introduce as many edits as required to rewire that cell so it becomes a living drug when introduced into the patient. Across our wholly owned and partnered efforts, we've generated a broad and diverse in vivo and ex vivo pipeline that's shown in this slide, with programs in the clinic or gearing towards the clinic and an expansive research stage portfolio, and I'll tell you more about it today. So for genetic diseases, we utilize our in vivo approach, where we systemically deliver the CRISPR machinery by IV infusion to the target organ, and our initial programs are liver-directed, to either selectively knock out a disease-causing gene or to achieve permanent gain of function with targeted gene insertion. And the goal is that we aim to develop these potentially curative therapies from a single dose. We're starting with the liver, but we're expanding to other organs. Recently, we talked about our efforts in bone marrow delivery by systemic administration. So maybe in the future I can come back and tell you more about those efforts. But today I will tell you about what we're doing in the liver. Core to the in vivo strategy is the use of lipid nanoparticles, or LMPs, for systemic delivery of the CRISPR machinery. For liver, LMPs are a clinically proven, safe, effective mode of systemic delivery of nucleic acids to hepatocytes. The drug format is shown on the cartoon on the left. We co-formulate a messenger RNA encoding Cas9 together with a target-specific single guide RNA in the same particle. As a platform, the only thing we need to change from program to program is the portion of the guide RNA that defines the target site for editing, which is shown here in different colors. For SPCas9, that is 20 nucleotides in the 5' end of the guide. The rest of the guide RNA, the scaffold region, the messenger RNA for Cas9, the composition, fabrication of the LMP, all of that is invariant. It's the same from program to program, and these are the benefits of platform approach. Now on the right, we're listing some of the key advantages of LMPs, particularly over other systemic delivery platforms such as viral delivery with AAV. LMPs have a large cargo capacity, so we're not size limited and can accommodate any type of editing tool. LMPs are well tolerated, they are non-immunogenic, there is no pre-existing or de novo immunity, so that doesn't limit patient eligibility. And if we need to redose or we want to redose, depending on the editing strategy, that is going to be feasible. And lastly, as chemical entities, they have scalable, controllable manufacturing, and this eliminates the cost and the complexity of viral vector production. After IV infusion, the LMP distributes to the liver, where it's taken up by hepatocytes via an ApoE LDLR dependent mechanism. So the LMP traffics to the endosome. As the pH drops, there is a conformational change in the lipid membranes that's mediated by protonation of the ionizable cationic lipid that results in a release of the cargo into the cytosol. So the mRNA gets translated into protein. Cas9 then binds to the single guide RNA that was co-delivered, forms a ribonucleoprotein complex, migrates to the nucleus that's facilitated by a nuclear localization signal on Cas, Once in the nucleus, Cas scans for the corresponding protospacer adjacent motif, or the PAM. Once it identifies the target site, only then Cas, there is a conformational change, becomes catalytically active, introduces the precise edit in the specific genomic location, and then there is endogenous DNA repair that comes and takes care of the edit. And for a knockout approach, the introduction of indels results in a frame shift that will lead to loss of expression of that gene. By delivering the CRISPR machinery in vivo, we can elicit several mechanisms of editing, and we're highlighting 3 of them here. We can knock out toxic or compensatory genes. We can insert or knock in therapeutic DNA sequences, whether it's for loss of function diseases, where we insert the wild type gene, or introducing a therapeutic protein, or a combination of both mechanisms that we call consecutive editing, or other mechanisms as required by the disease. Our first in vivo therapeutic program is Intellia 2001. Here we're aiming to permanently knock out a liver gene called transthyretin, or TTR. TTR is the causative agent for transthyretin-mediated amyloidosis, or ATTR, and this is a progressive, rare, fatal disease that's characterized by the accumulation of amyloid fibrils of TTR in tissues, predominantly in nerves, in heart, leading to polyneuropathy, cardiomyopathy, with many patients have a mixed phenotype. ATTR can be hereditary, and that's caused by over 120 different mutations that are spread over the gene. There is about 50,000 people worldwide with hereditary ATTR. The disease can also be acquired or wild type ATTR, and that is recognized as a cause for cardiomyopathy and heart failure in up to 500,000 patients worldwide. Both conditions cause debilitating symptoms and a high burden of disease. And although now there are approved therapies, chronic therapies for ATTR, there remains unmet need as patients are stabilized but not cured. Our goal is to halt and potentially reverse the cause of the disease by achieving deep, stable suppression of TTR. As a gene editing approach, once the gene is turned off, the expectation is that the effect is expected to be long-lasting following a single dose. Again, this is a cartoon depicting the drug candidate for TTR. So the drug product consists of a single guide RNA that targets the human TTR, and here is a target site that can lead to knockout not only of the wild type TTR, but also captures all the different mutations that cause hereditary TTR. In the same particle, we have the mRNA for the Streptococcus pyogenes, or spike Cas9 protein, formulated in a proprietary lipid nanoparticle, and this is the final drug product is a suspension for IV infusion. We've done extensive in vivo pharmacology studies in various models. I'll show you some of that data. CRISPR LMPs, as shown here, can lead to profound, widespread, homogeneous suppression in the expression of TTR in the liver. Here what we're showing is immunohistochemistry of mice that were untreated on the top, or at 1 week, or 6 months after editing. And as you could see that just in a week, you could turn off the expression of TTR in virtually every cell in the mouse liver. And that result is invariant when you're looking at 6 months after that single dose editing event. The deep stable suppression of TTR is associated with elimination of these amyloid deposits in various tissues, including nerves, as shown here in a mouse model of human mutant TTR. And you could see the deposits in the control group, and those deposits are eliminated after treatment. For nucleic acid therapies, non-human primates have really proven very valuable to inform on clinical translation. As shown here, we have a single IV infusion of the CRISPR LMP formulation, same formulation, same Cas as we would do in humans. The difference is that here we're targeting the monkey TTR for knockout. And as you could see, as compared to the untreated control, we had 3 cohorts of animals who got increased levels of the monkey TTR CRISPR LMP. And as you could see, a decrease in TTR serum is detected within a week after infusion of the LMP. It reaches a nadir by 4 weeks, and in this study, where we follow these animals for 1 year post-dose, those levels stayed suppressed, again, in variable results. Once the gene is shut down, it doesn't come back. These doses and the extent of editing that we achieve in these monkeys were what we used to project the clinical doses that I'll tell you about in a few minutes. In the non-human primates, we also perform ADME, so metabolism pharmacokinetics studies for the LMP, for the lipids, as well as the RNA cargos. And as shown here, and by design, all of the elements of the drug product are rapidly eliminated. And we like this. This is a desired attribute of our platform because we believe that it contributes to improved safety from the lipid and also from CRISPR, and the reduced risk of genotoxicity and immunogenicity. And, for everybody, Cas9, all the cases, right? This is a bacterial protein, so constant expression in your target organ could lead to cellular immunity. The ionizable cationic lipid of the LMP showed on the left, the guide RNA in the middle, messenger RNA, they're all eliminated within 24 to 48 hours post a single infusion. And this period is long enough to introduce the desired edits, as I just showed you on the previous slide. So I've been telling you about duration, and at least in the rodents and in the monkeys, we see long duration. To explore the potential to go beyond that, we use a model of accelerated liver proliferation called partial hepatectomy. So what we've done there is we edited so we had 2 cohorts, in teal the control, in blue a group of animals in which we edited the mouse TTR. So as you could see, after a week, you could get to 98% decrease in TTR expression. We took those animals, removed 2/3 of the liver, and now waited until the liver regenerated. And then the animals were sacrificed. And as shown on the right set of bars, we were very pleased to see that as the liver underwent very rapid rounds of proliferation, the cells that contributed to the growth carried with them the edit. So that gives us, again, an orthogonal information towards the potential for duration, right? So you're capturing enough precursors or, cells within the liver that can be edited by the LMP that will contribute to the homeostasis, the long growth potential of that organ. A very important aspect for the preclinical program for CRISPR is the identification, characterization, and selection of the guide RNA to ensure that it has high precision, specificity, no off targets. And just to make that clear, the goal is to identify a clinical guide that will potentially knock out the gene, in our case TTR, wild type, and all of the mutations, and that does not have any validated off targets at multiples of the intended pharmacological dose. For a canonical CRISPR-Cas9 approach, the target site that's defined by the guide RNA is what determines the entire landscape of potential off targets. As we just described, Cas9 only becomes active when it has identified the PAM and the guide RNA portion is properly hybridized to the genomic target site. So, knowing that sequence, it can help us now begin to query and using informatic tools for all the potential off targets, that will be perfect matches as well as with mismatches. So in addition of this computational approach, in the first phase of off-target analysis, which is the discovery of all these potential off-target sites, we combine this informatics approach with a biochemical and a cellular approach to try to understand all of the potential off-targets. And what we do is we aggregate all this information. We have all of lists with all the non-overlapping potential hits, and then we go one by one and demonstrate experimentally by doing dose responses in the appropriate cell type, which for liver is primary human hepatocytes from different human donors, and to look to understand whether these off targets are real, they're validated or not. And this validation is an incredible important part of the package that will go to regulatory agencies. For NTLA-21, we identified 7 validated off targets. All of them were in non-coding DNA, introns and intergenic regions. But very importantly, they were only observable at suprapharmacological doses. And these are doses that are not intended or likely achievable in the clinic. As shown in this dose response curve in a primary human hepatocyte, there were no detectable off targets at multiples of the intended pharmacological dose for TTR knockdown, which is here highlighted with the EC90. We started our global phase 1 study late last year. The first in-human study is a 2-part open-label multi-center study in patients with hereditary ATTR with polyneuropathy. We chose to start with patients with polyneuropathy because this is a population in whom a knockdown of the mRNA for TTR has been shown to have clinical benefit, and because this is the population that's most likely to provide clear data on safety while establishing a dose effect for NTLA-21. Part 1 is a single ascending dose part with a minimum of 3 patients per cohort. There are 4 dose escalation cohorts. The starting dose was chosen as 1/10 of the allometrically scaled dose at which we observed no adverse effect in monkeys. The optimal dose that will be selected in part 1 will then be administered to a dose expansion cohort in part 2 of the study. The primary objectives are to evaluate safety, pharmacokinetics, pharmacodynamics, and the pharmacodynamics includes serial measurements of TTR levels in serum, which is a direct readout of the editing that's happening in liver. In part 2, we will also begin to evaluate efficacy on clinical measures of neurological function. In June, we reported data on the first 6 patients from the first 2 completed dose escalation cohorts in the part 1. NTLA-21 was generally well tolerated, with AEs being all grade 1 and mostly unrelated to treatment. One patient had a mild infusion-related reaction, but it did not require dose interruption. And there were no severe AEs. There was no evidence of liver toxicity or coagulopathy, and laboratory excursions were insignificant. We observed dose-dependent serum TTR reductions by day 7. These reductions deepened by day 28. Here we have data from the 0.1 and 0.3 milligrams per kilogram cohorts. The reductions were 47 to 56% at 0.1 and 80 to 96 at 0.3. And as you could see here, there was good consistency in response among the 3 patients in each of the 2 cohorts. The average TTR reductions at day 28 were 52% and 86% respectively, with maximum reductions of 56 and 96% at 0.1 and 0.3. We published these interim clinical results with a very good description of the preclinical packages in June in an article in the New England Journal of Medicine. It's really good reading, including the supplementary material. So that was a landmark paper. This is the first demonstration of CRISPR-based in vivo gene editing in humans. It was a milestone for Intellia, for the team, for our partner Genome, for the field, but also very importantly for what this means for patients and the potential to tackling severe diseases with what could be potentially a permanent, curative new therapeutic modality. We continue to enroll patients across the sites in the UK and New Zealand, and we're submitting additional clinical trial applications as part of our ongoing global development for ATTR. And stay tuned for additional information on this program. We are really grateful to the patients and their families who participated in the trials, and also to the clinical investigators and everybody who have contributed to this important milestone. So what's next? With the clinical validation from NTLA-2001, it really strongly de-risks our in vivo platform. So now we can apply that platform modularity to other knockout targets and continue to build for our insertion targets as well. So the next program using this technology, this platform, is NTLA-2002 for hereditary angioedema, or HAE. HAE patients experience recurring, unpredictable, painful swelling in multiple tissues, and it could be lethal if untreated, particularly if they have inflammation, edema in the oropharynx area. They can asphyxiate. This is a serious disease. While there are acute and prophylactic therapies for HAE, there is a very high treatment burden, and breakthrough attacks still occur. So we believe there is an opportunity to have a therapy that will provide constant prophylaxis and the elimination of the attacks. So a bit on HAE and the mechanism. So HAE is a result of the dysregulation of the kallikrein-bradykinin cascade. And what happens is that the majority of these patients have an underlying condition, underlying deficiency in an inhibitor of this pathway called C1 inhibitor. As a result, this system is out of balance. And what happens is that now patients are hypersensitive to stimuli that normally will not result in angioedema. Kallikrein is a key mediator in the cascade. What it does is the enzyme that releases bradykinin from high molecular weight kininogen. So the therapeutic approach is to reduce the levels of kallikrein. That way we'll reduce the excess production of bradykinin with the goal of preventing vascular edema and resetting that dysregulated pathway. NTLA-2002 then aims to reduce the production of kallikrein, which is made by hepatocytes. And here we're targeting the prekallikrein gene that's called KLKB1. So by reducing the source of circulating prekallikrein, we can rebalance the cascade. We're excited about this program because, the therapeutic hypothesis is clinically validated. There is direct inhibitors of kallikrein. There is an antisense. Both of them have shown that reduction of kallikrein can reduce the attacks. So validating the hypothesis, but also informing on what's the target % inhibition that's required for an effect. Second, there are humans that are genetic nulls for KLKB1, and they do not have a deleterious phenotype. And third, as highlighted before, for this program, we're going to be leveraging our clinically validated liver-directed CRISPR platform, right? So whatever we saw for NTLA-2001, the projection is that will directly de-risk and inform this program. The only thing we changed from that 2001 to 2002 is the 20 nucleotides region of the guide RNA that now, instead of targeting TTR, targets KLKB1. For NTLA-2002, we conducted preclinical validation studies, again in multiple models. Here I'm showing data from a humanized KLKB1 mouse. The left figure displays the impact of 2002 on human KLKB1 gene editing in the liver. That's the red line. As the editing increases, we could see a decrease in the production, in the expression of the gene, measured by looking at serum kallikrein levels. And we observed a dose-dependent effect. The figure on the right now shows the pharmacology. That's the effect of 2002 on vascular permeability that is induced by a pharmacological agent called captopril. And captopril is an ACE inhibitor. ACE is the enzyme that will lead to bradykinin clearance. So by blocking it, we can augment bradykinin levels that will result in increased vascular permeability, and that we can measure with Evans blue dye extravasation. So upon treatment with captopril only, the TSS buffer, or with an irrelevant LMP, we could see an increase in vascular permeability relative to untreated animals, the baseline. And once that we treat with NTLA-2002, and we edit KLKB1, and we reduce the production of kallikrein, we can see a dose-dependent reduction in this vascular permeability. At the 0.3 milligrams per kilogram dose, which corresponds to about 85% reduction in kallikrein, there is a complete elimination of vascular permeability. Now, this study was done 14 days post-administration of NTLA-2002. To evaluate the durability of this effect, now we followed different cohorts of animals for up to 1 year after editing. And as shown by the data in this slide, the percentage editing, the decrease in kallikrein, and the pharmacological effect on vascular permeability remain unchanged from day 14 all the way to 1 year, which was the length of this study. The next pharmacology study was done in cynomonkeys using a surrogate guide RNA that targets the monkey KLKB1 gene. The graph shows data from 2 studies. Study 1 demonstrated dose dependence with maximum kallikrein activity reduction of 95% and durability to 1 year. The second study extended the durability assessment of the middle dose to 21 months. This is an ongoing study. Again, as shown for 2001, this supports the long effect of gene editing observed in rodents and non-human primates. The doses that we have tested here are doses that we expect are going to be achievable and safe and fully efficacious in patients. And for reference, that blue bar that's shown there is the threshold that we need to achieve based on the clinical data with other kallikrein inhibitors for gene editing to be efficacious. So the data is very encouraging, and assuming that this translates to the clinic, the therapy could potentially be a curative treatment for patients with hereditary angioedema. And we recently submitted our first regulatory filing to initiate a phase 1 study this year, and we're planning to be enrolling before the end of the year. We continue to deploy our toolbox to build our pipeline. One application is, in addition of knockout, is to precisely insert genes for gain-of-function indications. In this slide, we're showing data for insertion of Alpha1 Antitrypsin, the wild-type gene. This was done in non-human primates. As you could see by the graph, we were able to achieve normal levels of human Alpha1 in the monkeys, and these levels are stable through, here we're showing 30 weeks after insertion, after that single dose. And this is a study that continues. With our partner Regeneron, we're also developing insertion approaches for Hemophilia A as well as Hemophilia B. So we work hard to advance our in vivo platform to the point of clinical validation, and it now serves as a springboard to implement this long-term vision of developing additional candidates, not only in the liver, but continuing to unlock other tissues, including bone marrow. And that's a program that I'm particularly very excited about, the potential to do gene editing for sickle cell disease and beta thalassemia with a single IV infusion. More on that in future presentations. So now switching to our ex vivo programs, and here we're using CRISPR-Cas9 as a tool to create engineered cell therapies, and we're focused on reproducing natural cell physiology for improved safety and efficacy. Similar to the efforts that I just described in vivo, we have developed a proprietary cell engineering approach that serves as the foundation to the work that we're doing ex vivo. The approach allows us to mix and match cell types, targeting modality, and then the ability to introduce as many edits as required, insertions, knockouts, etc., with the goal of eliciting the desired pharmacology. Regardless of therapeutic format, we expect to achieve very high efficient editing, high yields, optimal cell performance, scalable manufacturing. And we have a sequential editing process that's with a proprietary platform technology. We maintain very high expansion potential of our edited cells as compared to commercially available processes. And we can do multiple edits, maintaining genomic integrity. So as shown on the right, we could do multiple edits by using a CRISPR-Cas9, and again, insertions and knockouts without having translocations or any other genomic rearrangements. Our lead program, NTLA-51, it has the potential to be a best-in-class therapy for patients with acute myelogenous leukemia or AML. And although there are some recent advances in AML treatments, particularly for some subtypes, the long-term outcomes continue to be poor, with less than 30% overall 5-year survival. It's a very tough disease. NTLA-5001, here we're using a TCR, a T-cell receptor approach. It recognizes a tumor antigen called Wilms tumor 1, which is overexpressed in the majority of AML, regardless of subtype. So this could be a universal AML therapy. With our proprietary cell engineering and the prevalence of WT1, we believe that this could be a really highly active and well-tolerated agent for patients with AML. NTLA-5001 utilizes our proprietary cell engineering process to replace the patient's T cells, endogenous TCR, with a natural high-avidity TCR that recognizes a peptide called VLD that's efficiently processed and presented broadly by AML blasts across, again, different subtypes of the disease. We're using our CRISPR engineering process, and what we do is, again, we take this is an autologous product, so we take the patient's T cells, we remove the endogenous TCR, create a blank slate into which we insert the therapeutic TCR. And by doing that, we achieve two things. The product is highly homogeneous because it only has that therapeutic TCR, so has high intrinsic potency. And by eliminating the endogenous TCR, you prevent mispairing of the alpha-beta chains of the endogenous TCR with the alpha-beta chains of the therapeutic TCR. And if that happens, now you could create these mispaired TCRs that will have unknown specificities and can lead to tissue toxicities. As shown by this efficacy study, 51 has a high potency against the tumor. And in this case, we're looking at a mouse model in which we use primary AML blasts from a patient to do this leukemia model. And as compared to untreated animals, or to animals in which they were treated with T cells displaying a irrelevant TCR, T cells with the WT1 TCR, that's the 5001, led to complete tumor growth inhibition. The proprietary cell engineering process that we have developed, it allows for rapid cell engineering, high yields, and these rapid vein to vein is really instrumental for autologous cell product. And as discussed before, the ability to replace the endogenous TCR with a therapeutic TCR leads to high potency and homogeneity of the cell product. Not talking today because of time constraints, but we have shown that through the process, the cells, they are not exhausted. They have a number of key attributes that we believe will translate into increased efficacy. We recently submitted our first regulatory filing to initiate a phase 1 study in patients with persistent or recurrent AML who have previously failed first-line therapies. And we expect to be screening patients by the end of the year. So ex vivo, we continue to invest in platform solutions for expanding our pipeline with additional immuno-oncology, autoimmunity, and future applications. Later this year, we're going to be talking about our proprietary allogeneic cell platform. Here, we're not only preventing graft versus host disease, but also elimination of the graft by the host T cells, CD4, CD8s, but also importantly by NK cells. And we believe that this is differentiated and will lead to really open up allogeneic therapies. And bringing it all together, at Intellia, we build these modular platforms that we're deploying in vivo and ex vivo. We continue to build and these robust toolbox that the goal is to be able to use the right modality for editing, with the right delivery to have medicines that are going to be transformational for patients. As I said in the beginning, the future is here today. This is a great time to be in this field and bringing the promise of nucleic acid therapies, particularly CRISPR, to patients. And I'm going to leave you with our 2021 strategic priorities and our guidance. And we're hiring, so if you were intrigued about what we're doing at Intellia, please visit our careers page or reach out to me. And I again thank you for the opportunity to present and look forward to the Q&A. Thanks a lot.
Laura, tremendous work with some really exciting data. I think I can safely speak for everyone when I say that I'm very optimistic for the future of this technology. So thank you again for sharing all this with us. We really appreciate it. To the audience, the Q&A is going to be coming up in just a few minutes. So if you have a question for Laura, please send it in. To do that, all you need to do is type it into the Ask a Question box and hit submit. Okay, since we're going to try to get to as many of your submitted questions as possible in the time that we have, we're going to transfer into the Q&A session. Bear with us for just a moment as we make that transition.
All right, everyone, thanks for joining us for the Q&A session. Laura, again, excellent presentation. Thank you so much for sharing all that information with us. And I'm going to take a little bit of moderator's prerogative here for the first question. At Gen, we like to keep up on latest news. So I know that Intellia issued a press release today with news about NTLA 5001. Maybe you could update us a little bit on that.
Sure. And, thank you again for hosting the webinar and for everybody who's participating and submitting questions. So today we had great news at Intellia because we announced our IND approval by the FDA for NTLA 5001. So as I was describing during the talk, this is our first ex vivo program, right, where we program in this case T cells for recognizing and killing AML blasts. So we have this approval by the FDA to initiate our first in-human study. We have guided that we're going to be, starting the trial with screening patients before the end of the year. So, this, continues to put, our guidance on track. We're super excited, and a bit more on the product candidate and on the trial that's, relevant to the news. So this is what we believe is going to be a differentiated, potentially best-in-class T-cell therapy, where we're using our Intellia engineering process to replace the endogenous TCR, T-cell receptor, in patients' T-cells with a high-avidity TCR that will recognize a tumor antigen, Wilms tumor 1, that is highly expressed and presented by AML blasts. And this is a universal, or seems to be a universal tumor antigen in AML. So it's going to be present across all the different subtypes. So, this is exciting because, for patients, because, today AML is a severe disease. And although there are drugs that have been approved for different subtypes, the overall survival is still poor. And, hopefully with 5001, we're going to have a, high active, safe, durable cell therapy that, that hopefully we can apply across all types of AML.
Thank you for that, Laura. I just got a note that we may have had a little bit of a lag in the beginning there. So for anyone who was just joining us, Laura was just updating us on Intellia's press release that was gone out this morning about news about NTLA 5001. So for anyone who came in a little late on that one, apologize about that. So Laura, we have another question from one of our audience members. They would like to know, with the validation of the platform, a natural question is what other indications do you see out there?
Yeah, so here we're talking now about the in vivo platform, where we're using the LMP, the lipid nanoparticle, to deliver CRISPR-Cas9 as the medicine systemically. And, the data that I just shared was on NTLA-2001, first demonstration of CRISPR activity in humans by systemic administration. So because of the modularity of our platform, now, again, as exemplified on the talk, when we can go to other diseases that are driven by genes that are expressed by hepatocytes, or where there are hepatocyte targets that play this role, a critical role in a disease pathway. And that's what we're doing for hereditary angioedema. And, the beauty of the platform approach is that, the only thing we need to change from candidate to candidate is a portion, basically 1/5 of the guide RNA, just the part of the guide RNA that targets the genomic location, that, genomic GPS for Cas to introduce the edit. So, with that in mind, and having all these platform attributes and elements that will translate to from program to program, it opens up additional opportunity in the liver. So, in the past, we have talked about other knockout targets that, we had been exploring, and these can range from rare diseases to prevalent diseases. And building on the safety that thus far has been demonstrated in the preliminary data from the ongoing phase 1, it really paves the way to have a well-behaved platform, again, that can be applied, all the way from severe diseases to high prevalent diseases.
All right, thank you for that. Our next question comes from Sonia, who'd like to know, is this process applicable to other types of ATTR, such as wild-type amyloidosis?
Yeah, thank you for the question, Sonia. So NTLA-2001 is designed to target both hereditary as well as wild-type ATTR. And the reason for that is that we selected the guide RNA to target a region of the gene that is conserved across all the different mutations and, of course, the wild type. So with that single guide RNA, we can turn off both wild type as well as any of the described mutations for TTR, and there are over 120 of them. So that's done at the onset of the program as part of the guide RNA design criteria. That in this case, we wanted to make sure that all patients, hereditary as well as acquired wild type, all of them could qualify to receive NTLA-2001 and for that candidate to be efficacious.
All righty. And our next question comes from Shaparno, who would like to know, how do you make sure that a good target of cells are edited with a single dose?
Yeah,
good number of target cells, sorry.
Yeah, I think you may be asking number of target cells. Yeah, so the lipid nanoparticles, you infuse them by IV infusion. It leverages an endogenous mechanism of targeting, which is this ApoE opsonization of the LMP that takes them to the liver, where this ApoE corona basically will be interacting with LDLR receptors in hepatocytes. It's a dose-dependent response, meaning, as you increase the dose of the LMPs, you could get to more and more hepatocytes in the liver. So at lower doses, you get to fewer hepatocytes. At higher doses, you could virtually get to every single hepatocyte. And there was, good demonstration of that was that immunohistochemistry in a mouse that I just showed that, basically every hepatocyte had that TTR turned off. So it's a dose-dependent response, and that's going to, so the dose selection is going to be also informed by what's the extent of target and knockdown that you would need to have, a pharmacological response. So we can, depending on what the target profile is, you could give more or less LMP. But the key point is dose dependent, and there are doses of LMPs that are safe, where you can get to virtually every single hepatocyte in the liver.
All right, thank you for that. Our next question comes from Wei Zhang, who'd like to know, what is the key item that caused safety concerns in toxicity studies? Was it LMP or RNA or expressed protein?
That's a great question. So, so as we're thinking about CRISPR candidates using a non-viral delivery system like the one I just showed, so you have two potential sources of toxicity, right, as you're outlining. One is delivery mechanism, two is the CRISPR mechanism. So for the delivery mechanism, the LMPs toxicities are very well characterized. They are in humans. There is an approved drug that is given to patients every 3 weeks. So we understand from that clinical experience that you could have LMPs that are very, very well tolerated. From the preclinical development of LMPs, we also, when you push the dose towards toxic doses, we also understand that these are dose dependent, they're monitorable, they're reversible. And the toxicities that you can see are, one is you could have an infusion type reaction. You could have increases in ALT because of cytotoxicity or toxicity to the liver cells. If the doses are high, you could begin to see also inflammatory responses and potentially coagulopathy. What's important is that as you're developing a human drug candidate, that it needs to work at doses that are absolutely safe, right, where you would not begin to see any of these dose-limiting toxicities. And and for NTLA-2001, what we've chosen to start in the clinic was with a dose that was more than 10x lower than the dose that in monkeys had no observed adverse effects. And that's part of how you think about drug development of these novel modalities. The second part of your question was about the cargo, right? So talking about can you see toxicity to CRISPR protein or to the RNA cargo? And we've designed our LMP and the mRNA to be everything to be rapidly metabolizing. It's there long enough to be able to introduce the edit, but then within 24 to 48 hours, the lipid components as well as the the RNA cargo and the protein that gets translated from the mRNA, all of that gets eliminated. So that brings together, again, improve safety, right? Because you're reducing the amount of material lingering in the liver, but also reduces any potential for immunogenicity or genotoxicity that could happen if, for example, you were to express, the Cas nuclease, which is a bacterial protein, for a long time. And you prevent having all of these potential issues by using a transient non-viral delivery system.
All righty, thank you for that, Laura. Next question from Sebastian, who'd like to know, could you please elaborate on the non-human primate to human delivery relationship you observed for NTLA 2001? Did the efficacy and off-target results translate well?
Yes. So when you go, when you're trying to come up with what are your human equivalent doses and what is the minimal starting dose, going from preclinical species to humans, you need to use allometric scaling that is traditionally used for drug development, right? To calculate what are going to be the human doses. So this is a very well understood methodology in clinical pharmacology, right? So to understand how to translate a rodent or a non-human primate to a human. So there is allometric factors that you apply to convert that monkey dose into a human dose, and this is what we had applied again to determine the minimal starting dose for the phase 1. As well as projecting the dose escalation that we projected to be fully efficacious. And thus far, our projections have worked really well for the doses that we selected for 2001, and we believe, again, because this is a platform approach, that they will translate to NTLA 2002 and other future programs in which we're doing a knockout mechanism.
All right, thank you for that. Next question we have is from Hossein, who would like to know, what is the size range of your LMPs? And goes on to say that, as you know, for the LMPs to get through the liver sinusoidal fenestrate, it needs to be less than 100 nanometers.
Yeah, and hi, Hossein. Thank you for the question. So, so that is absolutely right. So our lipid nanoparticles are below 80 nanometers. And what's also very important in addition of the size is that you have very low polydispersity, so that your drug product, all of the LMPs, all of the particles, they have very similar size and properties. But yes, it's important that you keep in mind the fenestration size across species and importantly in humans.
Okay, and a question here from Gary, who'd like to know, are there programs that are targeting non-liver organs? And what modifications to the LMPs are required for delivery to those organs?
That's an awesome question. So LMPs are particles. And this is something, as we were just talking about, that you need to keep in mind as you want to ensure or optimize delivery to other organs. It is true that the liver is, the perfect place for LMPs to go, but you can modulate the properties of the LMPs, and you could do that through chemistry and or process parameters that allow you to change some of the properties of that particle, so they can now access other organs, whether it is by systemic administration or applications that can be done by local administration. Last year, we presented data for sickle cell disease, or we're optimizing lipid nanoparticles to be given by systemic administration now. So we can access the hematopoietic progenitors in the bone marrow. This, the goal is that this is going to be a single IV infusion, a simple IV infusion in a patient. And those LMPs will make it to the bone marrow and be able to edit those progenitor cells, and of course that would have a number of applications. The obvious ones are for sickle cell disease and beta thalassemia. So stay tuned. We have shown that our LMPs work really well in a humanized mouse model. So this is in mice that have human CD34 cells. And stay tuned for more information on that program.
Okay, and our next question comes from Ishti, who'd like to know, is the in vivo CRISPR genome editing available for all therapeutic human diseases? How about degenerative diseases? Is that available too?
So that's the ultimate goal. The ultimate goal is to be able to deploy genome editing tools with the appropriate delivery modalities. So you could, aspirationally, right, go after any disease target gene in the appropriate cell type. Today, we are, in the beginning steps of that journey with demonstration by Intellia, right, in liver that we can target and the hepatocytes and turn off a disease-causing genes. Others are looking at other delivery modalities for in vivo use, for example, for ocular diseases. I strongly believe that through continued investment into what I call this toolbox of editing and delivery solutions, over time we will be able to address genetic diseases, for many other indications.
All right, and I think sort of following up on that, we have a question from Manish who says, LMPs can also be used in the case of neurodegenerative diseases, or can they be used in the case of neurodegenerative diseases? Please explain how the wrong cleavage can be prevented in eukaryotes as a major disadvantage, and how do you overcome this?
Yeah, so, for CNS diseases, right, let's start with delivery. So, you could think of delivery modalities, again, using a non-viral approach by local administration or intrathecal administration, as well as a viral delivery using, for example, an AAV. Regardless of the method, the other dimension that's important is selecting that guide RNA that's going to be absolutely specific for the target gene you want to edit with no off-target activities. And this is a design criteria that gets applied to every single program that, for CRISPR editing, that whether it's for in vivo use, liver or CNS or eye or any other organ, and also for ex vivo uses. So in the presentation earlier today, I showed you how we conduct upfront a very in-depth analysis of potential off targets. And the goal of that is to select a guide RNA that will not, and I underscore not, have off targets at the intended therapeutic dose plus a margin over that, safety factor over that. So that is the goal, right? So it's not to pick a guide RNA that will have off targets. It's pick a clean guide RNA from the start.
All right. And so, Laura, maybe could you talk about your vision for the company's next breakthrough in terms of technology platform?
So what's important for Intellia, we consider ourselves a genome editing company. And for us to keep that promise, it's very important that we continue to develop this toolbox of solutions. And the toolbox has gene editing modalities as well as delivery modalities, right? So the idea would be that we will be able to pick the right combination of what is the type of edit we want to introduce, and then have the enzymes as well as the delivery modality that will allow us to do that safely with the desired efficacy in humans. So, for us, with regards to delivery, for example, we have invested quite a bit in lipid nanoparticles. We have a, very experienced team looking, we have proven our LMPs, are safe and efficacious in humans. We're pushing the technology to go to other organs, such as bone marrow, I just described, and other applications. We also have a group looking at a viral delivery using adeno-associated viruses. What's very important there is that, as we do for LMPs, we want that expression to be regulated and transient. So that's, again, a design criteria, from the top. With regards to editing tools, we do have a variety of tools at our disposal, canonical nucleases, and we have ASP-Cas9, which is what we have in the clinic. We have a small Cas that, fits really nicely on an AAV for, that use or other uses. And then, we can use these two nucleases as the, as the basis to add different functionalities that allows us to do other editing modalities. And earlier this year, we talked about our proprietary base editing as an example of the tools that, we have or we're building.
All right, and I think we'll have time for one last question here, which our audience member asked, what are some of the key considerations when utilizing gene editing approaches to insert functional genes as compared to inactivating disease-causing ones?
Yeah, so and for that, I show you just one example on Alpha1 Antitrypsin. We're also developing factor 9 for Hemophilia B with Regeneron, as well as factor 8 for Hemophilia A. So what's important for liver now is, and the difference with the knockout programs is that now, in addition of delivering the gene editing machinery, we need to deliver a DNA template for insertion. Today, we're doing that with an promoterless AAV because we want to be the AAV to serve only as a delivery mechanism of the DNA for insertion, not to have any expression whatsoever, right, by itself. So, that's key difference, right? Now you need to have two elements and understanding, what is the appropriate level or dose of each of those components that will give you the right level of expression. We had shared before that we have, for example, for secreted proteins in the liver, we use the albumin locus for insertion. And in rodents, we show that there are three levers that we can use to regulate or to achieve different levels of expression. One is exactly where we insert the gene. The second is the dose of the CRISPR machinery by LMP, and the third is the dose of the AAV. So by doing a matrix, you could select what's the right proportion of elements that will give you the level of expression that you project is going to be efficacious in humans. So more on that, we will talk about sharing additional data on our research programs later this year. So I hope to be able to share more information.
All right, thank you very much, Laura. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website at genengnews.com. So if you missed any parts of it, feel free to watch it again, or you can forward it along to any of your friends or colleagues who you think might be interested in. I'd like to thank Laura again for her wonderful presentation, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to TriLink Biotechnologies for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.